Now that we've had a chance to go into the details of action potentials and how those work to send the signal down the length of an axon, we need to look at how we get that signal from the axon to the next cell. So we need to look at the connection between two neurons. The connection between a neuron and its target cell is a synapse. And in this case, we're going to look specifically at a synapse between two neurons. We start with the presynaptic neuron. The presynaptic neuron is the one that just had the action potential and is going to be sending the signal to the next cell. In order to do this, the presynaptic neuron has to release a neurotransmitter. So the electrical signal of the action potential that races down the axon of the presynaptic neuron is going to cause the release of a chemical. So we're actually going to have a chemical signal going to the next neuron. When we look at the axon terminal of the presynaptic neuron, we can see that it's filled with these vesicles. These are called synaptic vesicles, and they're filled with neurotransmitter, the chemical that's going to be released by the presynaptic neuron. The neurotransmitter gets released into the synaptic cleft. That's the space in between the presynaptic neuron and its target cell. In this case, the target cell is another neuron. We call this one the postsynaptic neuron because it comes after the synapse. The postsynaptic neuron has the receptors for the neurotransmitter so that the neurotransmitter can bind to the receptors and it can cause a change in the membrane potential of the postsynaptic cell. The axon of the presynaptic cell can form a synapse on a dendrite or the cell body, or rarely even on the axon of the postsynaptic cell. Although most typically we see that the connections go from the axon of one cell to the dendrite of the postsynaptic cell, and that's what's shown here. Let's take a closer look at the neurotransmitter. There are many, many different neurotransmitters that can be released by a presynaptic neuron to cause an effect in their target cell. This slide just shows a few of them. Again, the neurotransmitter is the chemical signal that's released at a synapse. In some cases, we can see the exact same molecule being released as a hormone when it's released into the blood instead of at a synapse. For example, epinephrine can be released into the bloodstream as a hormone, or it can be released into a synapse as a neurotransmitter. Another neurotransmitter we've talked about already is acetylcholine. Acetylcholine is the neurotransmitter that's released by motor neurons at the neuromuscular junction in order to trigger contraction in a muscle fiber. Other neurotransmitters you may have heard of before include dopamine. Dopamine is largely found in the brain, in the central nervous system, and dopamine is important for a number of things, including feelings of pleasure, which is why dopamine is often associated with addiction. Anything that causes the release of dopamine triggers an increased desire for that substance because it does create the feeling of pleasure. Dopamine is also important for helping to regulate movements, so some of the signals going from the brain to control the movements of our body use dopamine as the neurotransmitter. Serotonin and endorphins are neurotransmitters that are also associated with feelings of happiness and well-being. Endorphins are released in the brain as a result of excitement or exercise, which is one of the reasons people talk about something like a runner's high or about feeling so much better after they exercise. That's caused by the release of the endorphins, which also have the effect of reducing your perception of pain. Neurotransmitters can be either excitatory or inhibitory, meaning they can cause a depolarization in the postsynaptic cell or a hyperpolarization in the postsynaptic cell. This is not determined by the neurotransmitter itself, but rather by the type of receptor that the neurotransmitter is going to bind to. If the neurotransmitter binds to a receptor that is going to open sodium channels, then we're going to be depolarizing the membrane as sodium comes in, and that's going to be acting as excitatory at that particular synapse. On the other hand, the very same neurotransmitter released at a different synapse might bind to a receptor that opens potassium channels, causing potassium to leave the cell and hyperpolarizing the membrane. So whether a neurotransmitter is excitatory or inhibitory doesn't depend on the neurotransmitter itself so much as on the effect of the receptor that the neurotransmitter binds to in the postsynaptic cell. Let's take a look at the steps involved in actually getting the signal from the presynaptic cell to the postsynaptic cell. We know we start with an action potential in the presynaptic cell. The presynaptic cell got some sort of stimulus that triggered an action potential that races down the axon to the axon terminal. What happens in the axon terminal depends on calcium. We have voltage-gated calcium channels in the membrane of the axon terminal. And when the action potential, this big change in membrane potential, reaches the terminal, reaches those voltage-gated calcium channels, it causes those channels to open. When the voltage-gated calcium channels open, calcium comes into the cell. Calcium is what actually triggers the release of the neurotransmitter. It stimulates the process of exocytosis that releases the neurotransmitter into the synaptic cleft. The neurotransmitter diffuses across the synaptic cleft and then binds to the receptors on the postsynaptic cell. When the neurotransmitter binds to the neurotransmitter receptor, we're going to be opening or closing some sort of channel or changing some activity inside the cell that's going to adjust the membrane potential of the cell. If we raise the membrane potential with a depolarization, then we say that was an excitatory effect. And if we lower the membrane potential, make it hyperpolarized, we call that an inhibitory effect. The postsynaptic cell is going to have to look at all of the various local potentials caused by all of the different things it can detect in order to see if it sums up to the threshold potential, that magic minus 55 millivolts, at the axon hillock. If it does, then the postsynaptic cell will be triggered to have an action potential and send the signal to its target cell. Finally, as like almost everything we've looked at, we want to have a signal, but we don't want that signal to stay there forever. So once the neurotransmitter has been released by the presynaptic neuron, we need to get it out of the way again. We don't want that sitting there signaling indeterminably. We need to stop the signal. And we have a couple of different mechanisms for stopping the signal once the neurotransmitter has been released. One way to clean up the neurotransmitter is with the presence of enzymes in the synaptic cleft that can break down the neurotransmitter. For example, we saw acetylcholinesterase in the synaptic cleft of the neuromuscular junction to break down the acetylcholine after it was released. So we could break down the neurotransmitter with enzymes. However, it's nice not to waste the neurotransmitter, and so a second option is reuptake. There are actually pumps in the presynaptic neuron that take the neurotransmitter and pump it back into the presynaptic cell where it can be recycled and used again. The combination of the release of neurotransmitter and how quickly or slowly it's taken back up both have an important effect on how long the signal is going to be transmitted to the postsynaptic cell. If we can slow down the rate of reuptake, we can have a stronger or longer signal each time there's an action potential from the presynaptic cell. A specific case where this is important is in the case of serotonin. Remember that serotonin is a neurotransmitter that's related to feelings of well-being, and it's one of the targets of some antidepressant medications. A specific type of medication is called a serotonin reuptake inhibitor. The way this works is that the presynaptic cell signals and releases serotonin into the synaptic cleft. But if it gets cleaned up too fast, there's not a big effect of that serotonin and you don't have that feeling of well-being. By blocking serotonin reuptake, when the serotonin is released into the synaptic cleft, it stays there longer and it has a bigger effect on the postsynaptic cell, increasing feelings of well-being.